Everybody, and thank you very much for attending my talk today. I'm very sorry that I couldn't be there in person for personal reasons, but very glad to share some of our recent work on solid tumor targeting using NKG2D based CAR T cells, albeit virtually. I'd like to also thank Dr. Sonam for the very kind invitation to attend the meeting, and these are some of my disclosures. I'm an academic at King's College London, but I'm also affiliated with a CAR T cell spin-out company called Lucid Bio, hence the use of both lucid and academic nomenclature for some of the CARs that I will tell you about. But all of these CARs are targeted using a natural receptor known as NKG2D, which has 8 distinct ligands, which are very commonly expressed across a spectrum of solid and hematological malignancies. So this is essentially an innate immune surveillance system for stressed cells. And one of the key triggers for NKG2D ligand expression by cells is DNA damage, which of course is a hallmark of cancer. Now, we are certainly not the first to develop CAR receptors directed against the NKG2D ligand family. In fact, this field was pioneered by Charles Sentman more than 20 years ago, who developed the orange CAR that you can see on the right of this slide, in which he fused the full-length NKG2D receptor to the endodomain of CD3 zeta. And so by this means, he generated a fusion receptor which could deliver an activating signal when a T cell that expresses that receptor engages any one of the 8 NKG2D ligands. Now, at first sight, this appears to be a first generation CAR, and this, of course, is a technology that we know is not clinically enabled. But in actual fact, this orange CAR, which I've listed here as NKG2D CD3 zeta, is de facto a second generation CAR. And the reason for that is that this fusion of NKG2D and CD3 zeta will pair up naturally with endogenous DAP10, a co-stimulatory receptor present in T cells, and that complex then makes its way to the cell surface, delivering both an activating and co-stimulatory signal in response to recognition of these ligands. Now, this particular fusion receptor was developed by a company in Belgium as a product known as Syed-01. And Celiad Oncology is the name of this company, and they have treated more than 100 patients with CAR T cells engineered to express Syed-01 across a spectrum of both solid tumors and hematological malignancies. And because of this very extensive clinical experience, we have benchmarked the CARs that we have developed against an analog of the orange CAR, an analog of Syed-01. Now, Syed-01 was, I suppose, pretty similar to many CAR Ts in the solid tumor setting, demonstrating an acceptable safety profile, but relatively limited clinical efficacy. And so whilst we were very attracted to the target, the fact that these ligands are triggered by a lot of the stress events that happen in the solid tumor microenvironment, we thought that we needed really to go back to the drawing board in terms of designing the CAR in order to optimize potency and durability of function. And in an attempt to do this, we designed the blue CAR that you can see on the left of this slide, referred to here as NKG2D/DAP10-DAP12. This is an example of what we call an adapter CAR, whereby instead of having a linear fusion of the ligand binding domain of the signaling apparatus of the CAR, we instead are relying upon a non-covalent or salt bridge type association between the ligand binding domain, in this case NKG2D, and the signaling apparatus, which in this case is a fusion of full-length DAP10 to the endodomain of DAP12. I should mention that each component in this receptor has been optimized. In other words, we have found that use of DAP10 as the co-stimulatory domain in this receptor outperforms related analogs such as CD28, for example. And similarly, the use of DAP12 outperforms CD3 zeta. And we think the reason for this is reducing the dose of ITAM present per monomeric unit in the receptor. Of course, CD3 zeta has 3 ITAMs per monomer, whereas DAP12 has only a single monomer. Now, one of the differences that we found between T cells which express the cellular oncology analog, the orange CAR, and the adapter CAR that we have built, the blue CAR here, is the fact that we see higher cell surface expression of the blue compared to the orange CAR in transduced T cells. And to address that as a possible explanation for some of our results, we also built the purple CAR that you can see third along in this cartoon schematic, where we overexpressed additional exogenous DAP10 with the cellular oncology analog to create that structure there. And we also benchmarked against T cells that express just NKG2D alone as an additional control. So here you see the same 4 constructs on the left of this slide and some in vitro data on the right, demonstrating the ability of T cells which express these different receptors to undergo iterative restimulation on 2 different tumor monolayers, BXPC3 pancreatic cancer cells and Ren mesothelioma cells. And this is just a snapshot of a lot of in vitro data that we have generated and published quite recently, actually, showing that the blue adapter CAR can outperform all of the controls in these in vitro assays. We quite like these tumor restimulation assays because of the fact that we believe that they mimic the need for CAR T cells in the tumor microenvironment to undergo serial restimulation without the onset of exhaustion. So we tend to put a lot of faith in this particular assay. Next, we took these 3 constructs into an in vivo experiment using the same pancreatic cancer cell line, BXPC3, but established as a xenograft model. And if you just look at the orange CAR first, this is the analog of the cellular oncology CAR, we have essentially no efficacy here over and above what you see with PBS treated mice. And if you take that orange CAR and upregulate cell surface expression of the CAR by overexpressing DAP10, that's the purple CAR, you do not fix the problem. Whereas by contrast, with the adapter CAR shown in blue on the left of this slide, you are beginning to see some efficacy now, some degree of tumor control seen in maybe about half of the mice in this particular experiment. We then went on to test these constructs in a number of other solid tumor models. On top, we have a mesothelioma PDX tumor, which is a very different tumor actually to the pancreatic tumor I've just described, in the sense that it's a very slow-growing subcutaneous tumor xenograft. It takes over 100 days for the tumor to engraft in immunodeficient NSG mice. And what you can see in these mice, if you plot the volume of the tumor following treatment with the various CAR T or control therapies, indicated by the dotted line in each case, you can see that the cellular oncology analog, NKG2D CD3 zeta, shown in orange, has no efficacy, nor do NKG2D overexpressing T cells. They're both the same as PBS. Whereas with the adapter CAR, you can control the tumor completely in 6 out of 7 of the treated mice. The lower panels show 2 ovarian tumor xenograft models, and again, it's a similar story in terms of demonstrating the superior performance of the adapter CAR. You can see in the Kuramochi model that the cellular oncology analog, the orange CAR, has no efficacy once again, whereas with the blue CAR, you can achieve complete responses in 7 out of 9 mice. And if you re-challenge the apparently cured mice with a fresh injection of tumor on day 86, that re-challenge is rejected in 5 out of 7 cases. The lower tumor is another ovarian xenograft, A2780. Now, in this case, both the orange and blue CARs can achieve control of the tumor, but if you carry out a re-challenge on day 56 in both cases, then you can uncover a difference between the two treatments in that the cellular oncology analog CAR T cells fail to control the rechallenge, whereas the rechallenge is controlled in every one of the 7 rechallenged mice in the case of the adapter CAR. Now, one other attraction, I think, of NKG2D ligands from a conceptual point of view as a CAR T cell target, it relates to the fact that these ligands are not just expressed on the malignant cells in the tumor microenvironment, but they are also expressed on a range of different stromal cells that you find at that location, such as macrophages, myeloid-derived suppressor cells, regulatory T cells, and also mesenchymal stromal cells, as illustrated in this very simple 2D culture system. So what we've done here is to co-culture BXPC3 pancreatic tumor cells with pancreatic stellate cells. And what you can see are that the tumor cells grow as these islands, and I've put an arrow on one of the islands at the top, whereas the stellate cells form these long stromal elements. A very simple recapitulation of what you see in pancreatic tumors. Now, typically, when CAR T cells access a model such as this, they are naturally attracted to the mesenchymal stromal cells by virtue of chemokine receptor interactions. And of course, when those CAR T cells are stuck in the stroma, what they are not doing is attacking the tumor. But this affords no protection against NKG2D CAR T cells because even though they are initially attracted to the stroma, as you can see, they are rapidly destroying the stroma. And that's pretty much destroyed now. The tumor cell islands appear to coalesce in an apparent attempt to evade the CAR T cells, but this again affords no protection. And now you can see everything has been destroyed in this culture system. You can also carry out similar experiments in a 3-dimensional spheroid model where you mix together these tumor cells and mesenchymal stromal cells. They form this tight ball of cells, and then you can add in CAR T cells to the outside of these spheroids, asking the question, can they actually penetrate through and destroy the tumor and the stroma? And indeed, if you do the experiment with the analog of the SIAD-01 CAR, the NKG2D CD3 zeta fusion, to the naked eye, you really don't see any difference between the size of these spheroids and those treated with untransduced T cells. Whereas by contrast, with the adapter CAR, you can see that nothing remains visible to the naked eye. And if you analyze these cells in a flow cytometer, you can see that you can distinguish between the T cells and the tumor and stromal cells just on a side scatter, forward scatter plot. And here you can see actually that the Celyad oncology CAR does actually have activity. It certainly is reducing the content of tumor and stroma. And you can see in the middle panel that the T cells look more activated, indicated by a kind of a fatter scatter plot compared to the untransduced control. But by contrast, with the adapter CAR, there are really no residual tumor or stromal cells left. You've just got activated T cells, which are now beginning to encroach into that tumor plus stromal gate. Another important question that we needed to address regarding this adapter CAR, given the fact that this is quite a new structure, although it does recapitulate the structure formed by very many natural immune receptors, but another question that we were asked about this is how do you know that these adapter CARs actually coalesce or co-align at the cell surface? And we address this using 2 different approaches. On the right, you can see some confocal microscopy, which clearly demonstrates with the LU002 adapter CAR that both DAP10, NKG2D, and DAP12 are present at an identical location at the cell surface. I haven't shown it on this slide, but we have carried this out also with the Celyad oncology CAR. And what's quite interesting is that you see a lot of the NKG2D is actually trapped in the cytoplasm of these cells, suggesting that DAP10 is a limiting factor to get that NKG2D up to the cell surface. And the flow cytometry plots on the right simply once again show that we can detect both DAP10, DAP12, and NKG2D overexpression in these LU002 adapter CAR T cells. Another important attribute, we believe, of this adapter CAR T cell, LU002, is the fact that these T cells form highly organized immune synapses. I'm sure you're all aware of the fact that T cell receptors also form highly organized synapses with a central SMAC surrounded by a peripheral SMAC zone. So you have T cell receptor and some co-stimulatory receptors coalescing in the middle of this structure, surrounded by an actin ring in the PSMAC structure. Whereas by contrast, many traditional linear CARs, targeted most commonly, of course, by SCFVs, do not form this kind of highly organized bullseye structure. But we have found, as you can see in the bottom right of this slide, in some turf microscopy studies, that we do indeed see proper bullseye synapses formed by these NKG2D based CAR T cells. And this is something we are exploring at the moment in the context of what is driving this. Is it just the fact that we're using a natural receptor, NKG2D, to direct specificity, or has it got something to do with the adapter structure of this CAR? Now, one of the other interesting things that we noticed when we expanded these adapter CAR T cells in culture, as you generally do before you treat patients, is that we saw a very low expression of cell surface markers of senescence in these cells. So they express very low levels of KLRG1 or CD57, and that actually not shown on this slide was not seen with SIAD01 analog CAR T cells. Senescent T cells, on the other hand, typically downregulate the memory marker CD27, but we saw quite the contrast here in that we saw that these adapter CAR T cells maintained very high expression of CD27. So we then went on to carry out RNA sequencing analysis. This is bulk sequencing analysis, comparing our adapter CAR to CD3 zeta based linear CAR T cells. And once again, we saw downregulation of senescence associated gene sets in these T cells. So we believe that this is a feature of the adapter CAR structure, which is essentially contributing to the low level of senescence in these cultures. This gene set, this RNA sequencing analysis was also subject to gene set enrichment analysis, which uncovered two other interesting attributes of these adapter CAR T cells. Number one, an increase in gene sets associated with ribosome biogenesis, which of course is required for metabolic fitness of T cells as they tackle tumors. And we also saw upregulated gene sets associated with oxidative phosphorylation. And when we went back into the lab to do metabolic studies, this was borne out in the fact in the seahorse analysis that we saw an enhanced maximal oxygen consumption rate in these T cells and spare respiratory capacity, in keeping with increased oxidative phosphorylation capacity. So far, so good. We believe we have an NKG2D based CAR, which has nice anti-tumor activity in vitro and in some animal models. But of course, there is a very long way to go from those kind of studies to taking something into a clinical trial where you believe it may actually have a significant impact in the clinical situation. And one of the many additional obstacles that we were very conscious of in thinking about solid tumor CAR T cell immunotherapy is the fact that we have a drug delivery problem in this disease, in contrast to diseases like acute lymphoblastic leukemia, whereby you put the CAR T cells into the circulation and they have direct access to the disease cells. That is not the case with solid tumors, which originate from and metastasize to parenchymal organs. So how do you get your T cells to identify these areas of disease? And one of the approaches that we have been using to try to address this involves the armoring of this adapter CAR T cell product with a chemokine receptor, CXCR2. And so now we move in lucid speak from a product that I have referred to so far as LU002 to a composite product in which we co-express these transgenes, and this is referred to as LU011. The bar graph you can see on the right of this slide is a simple chemotaxis assay where we have interleukin 8 in the lower well of a transwell system, IL-8 being one of the CXCR2 ligands. And as you can see, this enables these LU011 T cells, shown as the dark blue bar, to traverse the transwell much more efficiently than the controls. And the lower panels simply show the co-expression of all of these transgenes in the T cells as we would expect. So, how do we know that this really actually enables these T cells to traffic into tumors? So, we initially addressed this in some in vitro studies with matrigel embedded pancreatic tumor spheroids. So, I'm going to run these three movies simultaneously. And what we see here is a spheroid growing in the middle of quite high density matrigel. We've added the T cells to the outside of the blob of matrigel, and the question is, can the T cells penetrate into the tumor in the center? So already 1.5 days into the culture, you can see that the Lu011 spheroid is growing more slowly than the other two. And by just over 2 days, you can see that the spheroid no longer exists. You just see this translucent area in the center of the gel. If you now turn your attention to the middle panel, the Lu002 panel, you can see now that this spheroid is lagging behind the untransduced control. And again, as we hit the day 4 to day 5 mark, you will see a very similar transition whereby the center of the spheroid becomes completely translucent, meaning that there is no tumor there. And you can see a kind of a gray swirl around that area, which are in fact the Lu002 CAR T cells. If finally you look over on the left of the slide at the untransduced control, you can see how infiltrative these tumor spheroids actually are if they are untreated. We've also tested this in vivo, and here you can see two different models. On the left, we have mice with an intraperitoneal tumor burden, and we are administering either the adaptor CAR T cells alone, that's Lu002, or the CXCR2 armored version, Lu011, and we're administering the CAR T cells intravenously and then imaging the T cells, not the tumor. So the question is, can we see T cells in the peritoneal cavity, which is the site of disease? And what's very clearly evident from these images, particularly at the 72-hour time point, is that there is a very clear increase in the signal at the site of disease. And this is also evident, perhaps to a lesser extent, in the subcutaneous tumors that you can see on the right of this slide, particularly by the day 15 and day 36 time point, if you compare the first two columns. And if you quantify this, there is a significant increase in influx of the CXCR2 armored T cells into these subcutaneous tumor xenografts. Now, the mere delivery of T cells to a tumor, such as the tumors I've just shown you on the previous slide, may not be enough, because what could be happening is that the T cells are getting to the site of disease, but then they are all aligned on the outside of the tumor in a kind of immune excluded phenotype, which of course is no good. But we don't believe that's the case based upon this. This is just being a representative example of an RNA scope analysis of one of these subcutaneous tumors, where the small black dots indicate these Lu011 CAR T cells. And as you can see, they're very evenly distributed across the core of the tumor. So how well does this work in vivo in preclinical mouse models? Well, this is just one of several examples, experimental examples that we have undertaken to test this question. I've selected this particular example because I think it really nicely illustrates the difference in performance between these different CARs. So what we have here is a pancreatic subcutaneous xenograft, CFPAC1, which has been allowed to establish to a volume of about 200 millimeters cubed before we give the T cells or the PBS control intravenously. And then we track the tumor volume using caliper measurements on the left. And as you can see, the T cells engineered to overexpress just NKG2D, shown in purple, overlap with the PBS treated control mice, indicating an absence of any efficacy in these two conditions. If you treat with an analog of the Celiad oncology CAR, where NKG2D is fused to CD3 zeta, you move to the pink line where you have tumor control to day 50, followed by outgrowth of the tumor. If you treat with adaptor CAR T cells alone, Lu002, the green line shows that you can control the tumor to day 100, followed by therapeutic failure. Whereas with Lu011, where you armor with CXCR2, even though the tumor burden is actually the highest at baseline in these mice, that's the red line, this line is brought down to the x-axis because of complete responses in all of the mice, which are sustained out beyond day 150, leading to enhanced survival. This is perhaps my favorite example, but we have carried this out across this type of experimental approach out in a total of 13 different xenograft and PDX models, representative of a range of common adult onset solid tumors listed on the bottom of this slide. And what you can see here is the survival of these tumor bearing mice following treatment with the indicated products on the key to this slide. So PBS shown in black overlaps with treatment with untransduced T cells shown in blue. Whereas by contrast, if you move to the analog of the Celiad oncology CAR, that is the pink line, you can see that there has been a clear difference compared to the control mice. And if I had truncated this survival curve at around about day 80, it would have looked pretty good for the pink line, actually. But the problem is that if you monitor the mice over their natural lifespan, these responses are not durable. And you can see that ultimately by day 200 plus, virtually all of these mice have succumbed to tumor progression. If you treat with the adapter CAR, Lu002, that is the green line. And now we have a further clear step up in efficacy, leading to 50% long-term survival of the mice over their natural lifespan. And finally, if you move to the Lu011 product, the red line, this now moves to 85% long-term survival with a clear plateau in the curve, which is generally indicative of tumor eradication. Now, if this approach is more efficacious, you might argue, well, it's likely to be more toxic as well. Addressing the toxicity of this approach preclinically is not very straightforward because of the fact that human NKG2D does not see its ligands in mouse models, for example. You would need to do non-human primate studies, which I must say, as a clinician, I find a rather unethical approach, given that we have many human patients with advanced and terminal malignancy who are very anxious to participate in trials of experimental medicines. So what we did instead is to remodel our adapter CAR so that we replaced the human NKG2D binding element with the mouse binder. And so you can see that as the kind of mid-hued blue oval on the top of the CAR, shown at the top here. So every other element in the CAR is identical to Lu011 with human DAP10, human DAP12, human NKG2D transmembrane domain, and human CXCR2. And we are injecting these CAR T cells into NSG immunodeficient mice, which have a subcutaneous tumor of human origin, mimicking what we will do eventually in the clinic. And we administer the T cells at escalating doses, the highest dose of which was 20 million CAR T cells. And that is a very high dose to be giving a mouse. We benchmarked these toxicity studies against an analog of the Celiad oncology CAR, which is shown on the bottom left of this slide. Once again, we swap out the human NKG2D ectodomain for the mouse ectodomain, so now this receptor can see its ligands in the mouse. And we again administer escalating doses of these T cells, and we monitor the two things in the mice. On top, we monitor the body weight of the mice, and on the bottom, we monitor the behavior of the mice, which is expressed as a grimace score. I should also point out that all of these experiments were carried out by a blinded investigator. So the investigator does not know which T cells he or she is administering to the animals. And what you can see, if you look at the weight on top, is there is a dose-dependent increase in weight loss, such that the animals treated with 20 million CAR T cells either are an analog of the adapter CAR, armored with CXCR2, or an analog of the Celiad oncology CAR. The weight loss is identical. It's maximal by about day 3 or day 4, consistent with CRS, and it's fully reversible by day 7 to day 8. And if you look at the behavior score on the bottom, again, you see an overlapping change in behavior. And this is generally reflected in lack of or reduced mobility of the animals and some degree of hunching, but not sufficient to exceed a humane endpoint and warrant culling of the animals. On day 8, all of these animals were killed and major organs were analysed for evidence of on target off tumor toxicity, and no such evidence was seen. So, in fact, what we saw in these mouse studies is very reminiscent of what Celiad Oncology saw in the clinic, with dose dependent cytokine release syndrome being the only significant toxicity seen with their NKG2D based CAR T cell product. So this, I think, is my last slide and summarizes the phase 1/2 clinical trial that we have initiated this year to evaluate Lu-011 in patients with solid tumors. And I'll just briefly describe the trial design because the actual results of the trial are embargoed until the forthcoming SITC meeting. But we designed the study with two single patient cohorts receiving 100 and 300 million CAR T, followed by reversion to a 3+3 design with two cohorts that will sequentially receive 1 billion and 2 billion CAR T, the intention being to treat a minimum of 3 patients in these cohorts, followed by an expansion phase. As you can imagine, there are very, very many people to thank for all of the preclinical data that I have shown you today. I will pay particular mention to Mark Davis, who did a lot of the early work on the adapter core, Dan Larkham-Young, Jana Obajdin, and also G. Tan. And also a big thanks to our clinical operations and CMC departments at Lucid, who are now facilitating the transition of this product into the clinic. And on that note, I will stop and be very happy to take any questions that may have